Welcome to the deep dive. We're here to unpack complex research and really get you the core knowledge you need. Today, we're actually going super small, right down to the cellular level. We're exploring something that, well, it underlies some massive health problems we see today. This idea of pathological lipid accumulation.
That's right.
Right.
You know, we often think about fat on a bigger scale diet, weight.
Yeah, exactly.
But this is about what happens when fat, lipids specifically, end up inside our cells where they just don't belong. It goes from being essential to, well, dangerous.
Okay, so we're talking about intracellular failures inside the cell.
Precisely. Failures in how the cell stores lipids, how it breaks them down, or how it ships them out. We're looking at the microscopic quality control going wrong.
So for this deep dive, let's clarify which lipids we're really zeroing in on. It's not just one thing, is it?
No, not at all. There are three main classes causing trouble here. First, probably the most familiar, triglycerides.
The energy storage guys?
Yeah. Exactly. 3 fatty acids on a glycerol backbone. Then you've got cholesterol and cholesterol esters. Crucial for membranes, hormones, but toxic if they pile up.
Right.
And finally, phospholipids. These are mainly structural parts of membranes. You often see them as these sort of weird world structures called myelin figures when cells are dying, specifically in necrosis. It signals membrane damage.
Got it. So our mission today is to really walk you through the main ways these lipids cause problems inside the cell and what diseases pop up as a result.
Yeah, and we want you to understand the critical difference, between a fat overload that the cell might recover from and one that sparks this dangerous, irreversible inflammation.
So what's the core problem? Why do cells start hoarding this stuff?
Well, fundamentally, it's about imbalance. Homeostasis breaks down. The cell takes in or produces more of a substance than it can actually process or get rid of.
Okay.
For lipids, our sources point to 2 main scenarios. First, the cell just can't remove a normal substance adequately. Think too many calories leading to excess triglycerides, the cell can't export.
Right, overload.
And second, there's a failure to break down a metabolite, often because of a genetic defect, like an enzyme deficiency. This leads to those lysosomal storage diseases.
Okay, let's tackle that first mechanism, the overload one. The classic example seems to be triglyceride accumulation, or what's called steatosis.
Exactly. Steatosis, or fatty change, that's the term for this abnormal buildup of triglycerides, and it happens specifically within parenchymal cells.
Parenchymal cells, so the main functional cells of an organ, right? The workers.
Precisely. The hepatocytes in the liver, muscle cells, kidney, tubule cells.
And you mentioned the liver, but also heart, muscle, kidney. Why is the liver always the poster child for this?
Because the liver is the body's central hub for fat metabolism. It's constantly dealing with fatty acids coming in from your diet or from your own fat stores.
Okay.
It takes these fatty acids, repackages them into triglycerides, and then, crucially, it has to bundle them up with specific proteins, apoproteins, to make very low-density lipoproteins, or VLDLs.
Ah, the VLDLs. Those are the transport trucks, basically.
That's a great analogy. They ship the fat out to the rest of the body. So, steatosis happens if any part of that process breaks down. Too many fatty acids coming in.
Like from obesity or diabetes?
Right. Or the cell can't burn the fatty acids for energy efficiently, maybe due to toxins like alcohol. Or can't make those apoproteins needed for the VLDL trucks.
Like in protein malnutrition.
Exactly. If you can't build the truck, the cargo gets stuck in the warehouse. The triglycerides pile up inside the hepatocyte.
So it's a logjam in the liver's fat factory. And visually, this is quite dramatic under the microscope, isn't it?
Oh, it's incredibly striking. We usually talk about macrovesicular steatosis. That's where the small lipid droplets fuse together into one huge fat vacuole.
A giant bubble inside the cell.
A giant bubble, yeah. And it literally shoves everything else out of the way. The source material describes it perfectly. The nucleus gets squeezed into the displaced rim of cytoplasm.
Wow, just physically pushed against the cell wall.
Like a crescent moon. The cell's functional machinery is just, well, crushed by stored fat.
That image really drives home the cellular injury. So what are the main culprits causing this disruption? You mentioned a few.
Yeah, the causes are quite varied because fat metabolism is so complex. But we can group them. Toxins are a big one. Alcohol is the classic example.
How does alcohol do it?
It directly messes with mitochondrial function, so the cell can't burn fat properly. Plus, it actually promotes the creation of more fat lipogenesis.
Double whammy.
Right. Then there's protein malnutrition. Like we said, no building blocks for the VLDL export trucks.
Okay.
And then the big ones today, metabolic disorders, type 2 diabetes, obesity. They just flood the liver with fatty acids, overwhelming its capacity to cope.
And things like anoxia, lack of oxygen.
Yeah, hypoxia or anoxia also inhibits fatty acid oxidation. The cell needs oxygen to burn fat effectively.
It really highlights the connection to modern lifestyle diseases. Our sources emphasize two massive drivers in developed countries.
Absolutely. Number one is still chronic alcohol abuse leading to alcoholic fatty liver disease.
Right.
But catching up fast and possibly overtaking it is non-alcoholic fatty liver disease, or NAFLD.
The silent epidemic.
That's what they call it. And it's almost always linked to obesity, type 2 diabetes, and the whole cluster of issues we call metabolic syndrome.
So for potentially millions, a fatty liver isn't just a liver problem, it's a bright red flag for the whole metabolic system being under stress.
It's the cell screaming for help. But here's the crucial point about steatosis, the hopeful part.
Okay.
Generally speaking, it's considered reversible cell injury.
Reversible, how?
If you remove the underlying cause, if the person stops drinking heavily, if they lose weight, manage their diabetes better, the cell can actually mobilize those fat droplets. It can burn them or package them up properly again.
So the nucleus can pop back to the center?
Yeah, the cell architecture can return to normal and function is restored.
Right.
The injury can heal.
That's huge. Injured, but not necessarily permanently damaged.
Right, but that's triglycerides. When we shift to cholesterol, the story often becomes, well, less forgiving.
Okay, let's make that shift. Let's talk about cholesterol and cholesterol esters and these four syndromes you mentioned.
So cholesterol is different. It's not primarily fuel like triglycerides. It's structural.
For cell membranes, right?
And hormones.
Exactly. Vital for membrane fluidity, precursor for steroid hormones, bile acids. Because it's so important, but also potentially toxic in excess, its levels inside the cell are usually very tightly regulated.
So if we do see cholesterol piling up in the cytoplasm,
Yeah.
something has gone seriously wrong with that regulation.
Seriously wrong. It usually points to either a defect in breaking it down, defective catabolism, often genetic or excessive intake, combined with the failure of the cell's clearance mechanisms.
And our sources lay out 4 distinct situations where this happens. Let's start with the really big one, the one impacting cardiovascular health globally. Atherosclerosis.
Atherosclerosis, yeah, this is the textbook example of cholesterol mismanagement causing disease. It all starts in the walls of large arteries, like the aorta or coronary arteries.
What happens there? LDL cholesterol particles, the bad cholesterol, sneak past the endothelial lining of the artery and get trapped in the layer underneath, the intima.
Okay, they're infiltrating the vessel wall.
Right. And the body sees this trapped, often modified or oxidized LDL, as foreign. So it sends in the cleanup crew. Immune cells, especially macrophages.
The immune system's vacuum cleaners?
Kind of. But they get overwhelmed. They gobble up huge amounts of this cholesterol and cholesterol esters. And as they get stuffed full, they transform. They get that characteristic bubbly look under the microscope.
The foam cell.
That's the one. They look literally foamy. And when clusters of these foam cells accumulate in the artery wall, they form those early fatty streaks, the beginnings of an atheroma or plaque.
But it's not just about the physical bulk of these cells clogging the artery, is it? There's something more dangerous when they die.
That's the critical transition.
Yeah.
That's what turns atherosclerosis from just a narrowing pipe into an active inflammatory disease site.
Oh.
When these overloaded foam cells die, either through programmed cell death or just bursting,
Necrosis, yeah.
they spill their contents. All that accumulated cholesterol gets released into the extracellular space within the plaque.
And it doesn't just dissolve away.
No. This free cholesterol is poorly soluble in that environment, and it rapidly crystallizes.
Cholesterol crystals. I remember the description from the source, quite vivid.
It really is. Histologically, when pathologists look at these plaques in tissue sections, they see these empty needle-shaped gaps or clefts.
Empty.
Yeah, because the actual sharp needle-like cholesterol crystals get dissolved away during the tissue preparation process. But they leave behind their footprint, these distinct clefts.
Okay, so we know the crystals were there, and the crucial insight now is what those crystals do. They're not just passive debris.
Absolutely not. They are potent danger signals. When nearby living macrophages try to engulf these sharp crystals,
They try to clean them up, too.
they do. But the crystals physically damage the macrophage from the inside. Specifically, they rupture the lysosome, the cell's waste disposal bag. And this internal damage triggers a specific molecular machine.
The inflammasome.
Exactly. Specifically, in atherosclerosis, it's often the NLRP3 inflammasome. Its activation leads to the release of powerful pro-inflammatory signals, like interleukin-1 beta.
So the crystal itself acts like a trigger, setting off an inflammatory bomb within the plaque.
Precisely. It turns the plaque from a stable deposit into an inflamed, unstable lesion that's much more prone to rupturing. And that rupture is what causes most heart attacks and strokes. The cholesterol isn't just blocking, it's actively fanning the flames.
Wow. Okay, that's atherosclerosis scenario 1. Scenario 2 takes these foam cells somewhere else, entirely skin and tendons, xanthomas.
Right. Xanthomas are basically visible, localized collections of these same foam cells. Instead of being inside an artery wall, they cluster together in the connective tissue just under the skin or sometimes in tendons.
And they look like?
They often appear as yellowish bumps or nodules, sometimes flat patches. They're essentially benign tumors made of fat-laden macrophages.
So if someone has these, it's a signpost on their skin pointing to a problem with cholesterol metabolism systemically.
Exactly. They are a classic sign of hyperlipidemic state conditions where there's way too much lipid, especially cholesterol, circulating in the blood. This could be genetic, like familial hypercholesterolemia, or acquired. They're a warning sign from the body saying, I can't handle all this cholesterol.
Got it. Okay, scenario 3 sounds a bit more mysterious. Cholesterolosis in the gallbladder.
Yeah, this one's interesting. Cholesterolosis is defined by these focal accumulations of foam cells. Again, cholesterol-laden macrophages, but specifically in the lamina propria. That's the connective tissue layer of the gallbladder wall.
And they call it strawberry gallbladder.
Sometimes, yeah. If the deposits are widespread, they can look like little yellow specks on the red inflamed background of the gallbladder lining, apparently resembling a strawberry surface.
But the weird part here, according to the source, is we don't really know why it happens there.
That's the kicker. Unlike atherosclerosis, where we understand the LDL infiltration or xanthomas linked to high blood lipids, the exact mechanism driving foam cell formation, specifically in the gallbladder wall in cholesterolosis, is still labeled as unknown.
Huh. Any theories?
It's thought to be related to bile that's supersaturated with cholesterol secreted by the liver. But why the macrophages in the gallbladder wall specifically start taking it up and hoarding it isn't fully clear. It's usually found incidentally and doesn't cause major problems unless it contributes to gallstone formation.
A little medical mystery. Okay, final scenario, number 4, takes us firmly into genetics. Niemann-Pick disease, type C.
Yes, NPC is a devastating example of a lysosomal storage disease. It perfectly illustrates the second major mechanism we talked about, the failure to degrade, or in this case, transport a metabolite due to a genetic defect.
So what's the specific failure point in NPC?
The underlying cause is usually mutations in the MPC1 gene, sometimes MPC2. These genes code for proteins that are essential for getting cholesterol out of the lysosomes and late endosomes.
The cell's recycling centers.
Exactly. Normally, when cholesterol enters the cell, it gets trafficked to the lysosome for processing. The MPC proteins are like the exit doors, allow the cholesterol to move on to other parts of the cell where it's needed or can be esterified for storage.
But in NPC, the doors are broken?
Essentially, yes. The cholesterol gets delivered to the lysosome, but it gets trapped there, it can't get out. So it accumulates massively within the lysosome, causing it to swell up.
And this damages the cell.
Severely. This buildup disrupts normal lysosomal function and eventually leads to cell death. Because cholesterol handling is vital everywhere, you see this accumulation in many organs, liver, spleen.
And the brain too, right? That causes the neurological problems.
Critically, yes. The accumulation in neurons leads to progressive neurodegeneration, which is usually the most severe and life-limiting aspect of Niemann-Pick type C. It just hammers home how crucial even one step in intracellular lipid trafficking is for the health of the entire organism.
4 very different outcomes: atherosclerosis, xanthomas, cholesterolosis, NPC, all stemming from problems handling cholesterol inside the cell. Okay, so we've laid out the foundational pathology. We've seen reversible triglyceride issues like steatosis, and then these more aggressive cholesterol problems. Now let's connect this to what's happening today. How is this knowledge from, maybe decades ago informing the cutting-edge research and treatments now?
This is where it gets really exciting because understanding the why at the cellular level directly leads to the how of modern therapies. Take that simple fatty liver, steatosis.
Right, NAFLD. You said it was reversible, but it's become a huge problem because of obesity.
Exactly. The clinical concern isn't just the fat itself, it's a progression. When NAFLD progresses to NASH, non-alcoholic steatohepatitis, that's when things get serious.
NASH, what's the difference again?
NASH means you have the fat, steatosis, plus inflammation, plus evidence of liver cell injury, like ballooning hepatocytes. And crucially, Often fibrosis starts scarring.
Scarring, that leads to cirrhosis, right?
Potentially, yes. Cirrhosis and liver failure, or liver cancer. So, the big push in medicine now is stopping NAFLD from becoming NASH,
Sure.
or reversing NASH before the scarring becomes permanent.
For years, it was just lose weight, eat better, exercise. Important, but hard. What are the new therapeutic targets?
Researchers are hitting it from multiple angles, targeting the underlying metabolic chaos. One really promising area involves drugs that modulate the farnesoid X receptor, or SXR.
FXR agonists. Okay, how does fiddling with FXR help a fatty liver?
FXR is a nuclear receptor, mostly found in the liver and gut. It normally gets activated by bile acids. Think of it as a master metabolic sensor.
Okay.
When you activate FXR with one of these agonist drugs, it basically tells the liver cell, okay, stop making so much fat, start burning more fat, and handle bile acids better. It decreases lipogenesis, increases fatty acid oxidation, and also seems to have anti-inflammatory effects.
So it's trying to reprogram the liver's metabolism back towards normal.
Pretty much. And clinical trials have shown some FXR agonists can actually get rid of the inflammation and ballooning without making fibrosis worse. That's a major goal.
What about those diabetes drugs we hear so much about? The GLP-1s.
Ah, yes. The GLP-1 receptor agonists like semaglutide, liraglutide, they've been game changers not just for diabetes and weight loss, but unexpectedly for NASH too.
How do they work on the liver? Is it just the weight loss?
The weight loss is definitely a huge part of it. Less weight means less fatty acid flowing to the liver, less insulin resistance. That alone helps tremendously.
Makes sense.
But there's growing evidence they might have direct beneficial effects on the liver cells too, reducing inflammation, maybe even directly impacting fibrosis pathways, kind of independent of the weight loss. It's still being fully worked out, but the results in NASH trials have been very impressive.
It really shows how interconnected everything is: obesity, diabetes, liver health. Treating one helps the others.
Absolutely.
Yeah.
That steatosis isn't just a liver cell problem. It's a symptom of the whole system under metabolic duress. Fix the system, the cell can also recover.
Okay, let's pivot back to cholesterol and those nasty crystals and atherosclerosis. You mentioned the NLRP3 inflammasome. Let's unpack that a bit more. How exactly does a crystal flip that switch, and why is targeting it such a big deal now?
Right. This is central to the modern view of heart disease as an inflammatory condition, not just a plumbing problem. NLRP3 is part of the innate immune system, designed to sense cellular damage or stress, even non-infectious sterile danger signals.
And the cholesterol crystal is a perfect example of a sterile danger signal. Sharp, indigestible.
Exactly. So activating NLRP3 usually involves two steps, a two-signal model. Signal one is priming. Things like oxidized LDL or inflammatory cytokines get the macrophage ready by increasing the production of the inflammasome components, like the precursor form of IL-1.
So the cell loads the gun, so to speak.
Good analogy. Then comes signal two, the trigger. That's the cholesterol crystal. When the macrophage engulfs it, the crystal damages the lysosome inside the cell. This damage causes changes like potassium leaking out, which triggers the assembly of the NLRP3 complex.
And that assembled complex is the active inflammasome.
Yes, this complex then activates an enzyme, caspase-1, and caspase-1's job is to chop up that inactive pro-IL-1 into the active, highly inflammatory cytokine interleukin-1 beta, IL-1.
And IL-1 is the real troublemaker here.
It's a major driver of inflammation in the plaque. It attracts more immune cells, promotes more cell death, leads to more crystal release. It creates this vicious cycle that destabilizes the plaque.
That sounds bad. How do we know blocking this actually helps patients?
The big proof of concept came from the CANTOS trial. They used a specific antibody, canakinumab, that neutralizes IL-1 directly.
What did they find?
They gave it to people who'd already had a heart attack and were on statins, so their LDL was controlled, but they still had signs of inflammation. The canakinumab group had significantly fewer recurrent heart attacks, strokes, and cardiovascular deaths compared to the placebo group.
Even with good cholesterol levels?
Even with good cholesterol levels. It proved that targeting the inflammation itself provided additional benefit on top of lowering LDL. It fundamentally shifted the paradigm.
So it's not just about the cholesterol number, but how the body reacts to the cholesterol that's there.
Precisely. It validated inflammation as the key therapeutic target in cardiovascular disease. It's also why older drugs like colchicine, which has some anti-inflammasome effects, are now being used successfully to reduce cardiovascular risk. We're treating the fire, not just the fuel.
Amazing. Okay, one more area of advancement. Let's loop back to Niemann-Pick type C, that genetic disease where cholesterol gets trapped in the lysosomes. What's new there?
NPC is tough, especially the neurological decline, since the core problem is that faulty NPC1 protein leading to trapped cholesterol. Treatments try to work around that.
Gene therapy, maybe? Replacing the faulty gene?
That's the ultimate goal, and work is ongoing, but it's complex, especially for the brain. In the meantime, there's been a lot of focus on a compound called cyclodextrin.
Cyclodextrin, what is that?
It's basically a type of sugar molecule, an oligosaccharide, shaped like a ring or a cone. And it has this unique property. It can bind to cholesterol. It acts like a molecular shuttle or sponge.
Okay. So how does it help in MPC?
The idea is that cyclodextrin can get into the cells, even cross the blood-brain barrier to some extent, and literally pull the trapped cholesterol out of those overloaded lysosomes. It essentially provides an alternative exit route, bypassing the need for the faulty NPC protein.
It chemically extracts the stuck cholesterol.
Exactly. It helps mobilize it. It's not a cure, but studies, especially where cyclodextrin is given directly into the cerebrospinal fluid intralathically, have shown it can slow down the progression of the devastating neurological symptoms in some NPC patients.
Are there other approaches too, like reducing the amount of stuff going into the lysosomes?
Yes, that's called substrate reduction therapy. For example, the drug miglustat is sometimes used. It doesn't directly target cholesterol, but it inhibits the synthesis of other lipids, like glucosylceramide, that also accumulate in NPC lysosomes. The idea is to lessen the overall burden on the dysfunctional lysosomal system.
So really targeted strategies based on knowing exactly where the molecular machinery broke down.
Absolutely. Understanding the precise defect allows for these kinds of rational therapeutic designs, trying to either compensate for the defect or reduce the load on the broken pathway.
Okay, what a journey. We've gone from, simple fat droplets in liver cells all the way to complex gene defects and cutting-edge drugs targeting inflammation. Let's try and wrap this up. What are the big takeaways for someone listening?
I think the main thing is to recognize those two broad categories of problems we discussed.
Yeah. First, the triglycerides leading to steatosis or fatty change.
Yeah. Driven mostly by metabolic overload diet, lifestyle, associated conditions. And the key here is that it's often reversible if you tackle the root cause.
Okay, category 1, reversible triglyceride overload.
Then category 2, cholesterol and cholesterol esters. This is where things get more complex and often more dangerous.
Because the accumulation is due to deeper issues like transport defects or problems breaking it down.
Exactly. And this leads to diverse, often systemic, and frequently irreversible pathologies. The prime example being atherosclerosis.
Where it's not just the cholesterol, but the inflammation triggered by its crystals that drives the disease.
Precisely. And you also have the xanthomas and the specific genetic storage diseases like MPC.
So the critical difference really comes down to how the cell handles the lipid and what the consequences are. Is it just storing excess fuel, or is it dealing with something inherently disruptive, like a crystal that ignites a bigger fire?
That's a perfect way to put it. Is it a contained storage issue, or is it actively promoting inflammation and damage? That distinction often determines whether the cell injury stays local and potentially reversible, or whether it contributes to chronic, progressive, life-threatening disease.
It really drives home that cellular health is systemic health. What happens inside one tiny cell, multiplied millions of times, dictates the health of the whole organism.
It's a direct reflection. The state of your cell's internal lipid environment mirrors your overall metabolic health.
Which brings us to a final thought, something to maybe chew on. We briefly touched on this pigment called lipofuscin.
Ah, yes, the wear and tear pigment. Pathologists see it building up in cells of older individuals' heart muscle, neurons, liver cells.
And what is it made of?
It's basically cellular garbage aggregates of cross-linked, oxidized proteins and, significantly, lipids. It's thought to be the end product of years of damage from free radicals, particularly damage to cell membranes through lipid peroxidation.
So it's like the accumulated rust of cellular life, partly derived from damaged fats.
In a way, yes, it doesn't seem to be acutely toxic itself, but its presence signifies a long history of oxidative stress and incomplete cellular housekeeping.
So if part of aging involves the slow accumulation of lipid-derived damage, it makes you wonder, doesn't it?
It poses a fascinating question. If we get much better at helping ourselves manage lipids throughout life through diet, maybe through future therapies, targeting metabolism or inflammation even more precisely.
Are we just treating specific diseases like fatty liver or heart disease?
Or are we potentially doing something more fundamental? Could improving cellular lipid handling actually slow down that process of accumulating wear and tear, Could it influence the biological rate of aging itself?
The battle against fat inside the cell might just be part of the battle against time itself. Definitely something to think about.
Indeed. Keep exploring the science that underpins how our bodies work. It's endlessly fascinating.